How to use a micropipette correctly. Micropipettes are expensive and a vital part of the laboratory. Proper care must be taken as they are expensive. Micropipette must be used correctly to get an accurate result. Note: using too much force to turn the control button outside the specified range may cause the micropipette to jam and become damaged. The range of this micropipette is from 100 microliter to 1000 microliter. Do not turn the control button below 100 microliter and above 1000 microliter. When you are either very near to 100 microliter or 1000 microliter, turn the control knob slowly and cautiously. Pre-rinse the tip 3 to 5 times when dispensing viscous liquids. Pre-rinse the tip 3 to 5 times when dispensing liquids that are at a different temperature than room temperature. The two most commonly used pipetting techniques are forward technique and reverse technique. Forward technique. This technique is commonly used for pipetting aqueous solutions such as buffers. It is also used for mixing reagents or pipetting sample into the reagent. Reverse technique. The reverse technique is used for pipetting viscous and foaming liquids. It is also used for pipetting whole blood. I am going to give demonstration on how to use micropipette correctly. Micropipette has two main parts: the control button and the ejector button. Control button has three positions. Now the control button is at resting position. It always comes to resting position when you release the control button completely. When you press the control button, it has two stops: the first stop and the second stop. While drawing liquid, always hold the pipette vertically. I am using three colored liquids, 1 and 2 as reagent and 3 as the sample. I am also using a wide mouth vial to make the video clear. Insert the tip into pipette by pressing it firmly. I am setting the micropipette to 1000 microliter for reagent 1. Press the control button to first stop. Hold the pipette vertically and immerse the tip into the reagent as small as possible, about 2 to 3 mm. Release the control button. While taking out the tip, slide it at the edge of the container. While dispensing, tilt the pipette slightly. Press the control button to first stop and press it further to second stop to empty the tip. While taking out the tip, slide it at the edge of the container. Release the control button and eject the tip into tip waste container. I am setting the micropipette to 500 microliter for reagent 2. Similarly, pipette reagent 2. First, touch the inner wall of the container with the tip. While dispensing reagent 2, let the tip not touch the reagent 1 which is already in the receiving container. Mix it well. I am going to add sample into it by forward technique. In forward technique, you push the control button to first stop. Immerse the tip as small as possible and release the control button. While taking the tip out, slide it at the edge of the container. Holding the pipette vertically, immerse the tip into the reagent as small as possible. Press the control button to first stop and press it further to second stop to empty the tip. Keeping the control button in the second stop position, take out the tip from the reagent. Do not draw back the reagent into the tip. While taking the tip out, slide it at the edge of the container. Release the control button to resting position and eject the tip. Mix it well. Out here, I have added reagent 1, reagent 2 and sample into it by forward technique. I am going to demonstrate how to pipette whole blood. First, let me add reagent into these two vials. And later, I will pipette blood into these two vials using two different techniques. The whole blood is a heterogeneous mixture of both solid and liquid components. The liquid part is called plasma, while the solid part consists of red blood cells, white blood cells, and platelets. I am going to demonstrate how to pipette whole blood by reverse technique. Always shake the blood well before pipetting. In reverse technique, press the control button to second stop. Dip the tip into the blood and draw the blood. While removing the tip, slide it along the wall of the container. Wipe the outer surface of the tip with tissue paper. Immerse the tip into the reagent. Dispense the blood into the receiving container by pressing the control button to first stop. Hold the control button in the first stop position and remove the tip from the reagent. The remaining blood in the tip is dispensed back into the original blood by pressing the control button to second stop. Release the control button and eject the tip. And mix it. This is another technique to pipette the whole blood. Press the control button to first stop. Immerse the tip into the blood and draw the blood by releasing the control button to rest position. While removing the tip, slide it along the wall of the vial. Wipe the outer surface of the tip by tissue paper. Dispense the blood into the receiving container by dipping the tip into the reagent. While keeping the tip in the reagent, release and push the control button 2-3 times until the tip is clear of the blood. Take the tip out of the reagent by sliding it along the wall of the container. Then press the control button to the second stop to empty the tip of any remaining liquid. Release the control button to rest position and eject the tip. Shake it well.